I can restart. I always say the same things. No, it's actually not true. I say random things usually. So I started out PhD in pharmaceutical technology. Pharmaceutical technology are the boring things like how you make a tablet. I didn't start out with how you make a tablet. I started out with how you make nanoparticles, nano-sized drug carrier systems. Then I gave my precious preparation that I spent months on making nanoparticles with a new drug. I gave them to people who tested them in cell culture or in animals, and they didn't treat them well. They did awful things to them the first time I did that. People told me afterwards, oh, we sterile filtrated them. And that means you put them through a filter, through a very tiny filter, and you filter out all bacteria. The problem was they also filtered out all of my precious nanoparticles, so they didn't have any effect. So I thought I have to learn the other thing, and I did a postdoc, and I moved to the university clinics in Frankfurt to do a postdoc, and that is where I started to work on viruses and cancer. I always thought I would come back to pharmacy at some point and do these things again with all the knowledge I have about the preclinical model systems, in particularly cell cultures, but I never did. I actually ended up in a school of biosciences now, so I'm full cycle in what you can do in this, I don't know, drug-related, disease-related research, pharmacy, medicine, and now biology. And I've always been working on cancer and on viruses, but today we're going to talk on cancer. And you can make the most out of it, and we can discuss a lot. I will ask you questions, and you can ask everything you want. Obviously, there's always the risk that you catch me out, and there are things I don't know, but I will be honest. If I really don't know something, I will tell you. But I also know the one or the other thing. If you're not that interested in talking with me and not having many questions, I have many slides, and we can know if I can take you through. I will be very excited because then I talk even more about my research, but I'm not sure how excited you will be. All right, what is cancer? What are the two features that identify a cancer cell?
Cells that are multiplying faster than they should be.
So that's what you read in the textbook and in the newspaper and everywhere. Cancer cells divide faster. That is not always clear. If you think about it, it's even very difficult to work out how fast cells are dividing. It's not difficult if I have a cell culture dish here, but it's very difficult in me as a person. But the idea is basically, but there is some truth to that, because we know that in fast-growing cancers, the cytotoxic therapies, the chemotherapies that we use, are the most effective ones. So there is something behind that. But more precise is to say uncontrolled growth. Cancer cells are cells that are in a tissue context, and they start growing and forming a lump, although they shouldn't. And the other basic feature of cancer cells, so one is uncontrolled proliferation. What else do cancer cells do? Have you ever heard of a disease called cancer?
The mutations that accumulate means that they have the potential to pass on and affect normal healthy tissues and destroy it completely, in the case of unregulated proliferation cancer cells.
Yes, so the mutations are very important. Mutations are a very important driver of cancer. And they are more an enabling factor. They enable cancer cells to do what they do. This is not a regulated process. That's very important. These things happen completely by random. But I think you also mentioned the second bit, and that is that cancer cells can move around. Cancer cells show invasive behavior. So if if a cancer starts in the prostate, sooner or later you will find it in other tissues. Many bone metastases in prostate cancer. Colorectal cancer usually metastasizes first into the liver. So the second essential feature of cancer cells is that they show invasive behavior. They don't respect tissue and organ barriers. They start to move around. So that's basically how I would define cancer. Cancer is an genetic and epigenetic disease. Genetic are all changes to the DNA, mutations basically. Epigenetic means changes to the way in which the DNA is expressed. That means which genes are active and which aren't. But it's not encoded in the code of the DNA. But nevertheless, these changes can be inherited, at least via one cell division. Sometimes such epigenetic changes can be inherited from mother to child. Yeah, they can go over generations by at least one cell division. And these are typically chemical modification of the DNA directly, or proteins that are involved in keeping the DNA in shape. So we have these genetic and epigenetic changes, and they result in a reprogramming of, actually, I don't know whether that's quite correct, in a reprogramming of cells in a way that they start to divide, although they shouldn't, and they start to move around, although they shouldn't. So that is cancer. And that is what all cancer diseases have in common. You can have, that's really a funny thing, isn't it? But it happened twice already. There seems to be a concept behind it. So in every tissue in an organism, you can develop cancer. It's not a topic for hypochondriac people, actually. No health topic actually is. So yes, what was I going to say? I don't know, it's written there. Oh yeah, in every tissue you can develop cancer. In some tissues it's much more common, in others it isn't that common. But the principles, the basic features of cancer cells are always the same. So my next question is, how can cancer be cured? And that will also lead us to the next question, or the second question. Well, we have already answered the first question of my talk, what is cancer? We know now. The second question is, why is it so difficult to cure? And so how can cancer be cured? Do you know about one cancer? Do you know which proportion of cancer cases is actually cured in a country like the UK? How many people who are diagnosed with cancer are actually cured? Which percentage? A brave guess by anyone?
I'm not sure about an average, but I think pancreatic cancer in the UK specifically was like, I think I can remember was only about 5% are actually cured.
Yes, pancreatic. Yeah, so it's obviously there's a huge difference. That's true. So there are cancers that are hardly ever cured. And pancreatic cancer is potentially the worst of them all. There are some others who are really, really bad. But do you know, on average, so of 100 people who are diagnosed, yes? 50%? Yeah, that's about right. About half of people who are diagnosed with cancer are actually cured. That means the cancer basically cured. It's an interesting question. We don't basically know 100% they are cured. What we think means cured is somebody has had cancer, was treated. We don't detect any cancer in their organism anymore, and they have died from something else. So they didn't have a recurrence of the cancer. So it may well be that some people, if they had lived 50 years longer, they would have had a recurrence of their cancer. So there are many things we don't know. So how are most people, what can you do to cure a cancer patient?
You can do a general treatment, which is like the chemotherapy or radiation that will kill fast-growing cells, although it tends to be quite dangerous for the patient's healthy cells as well, because they are just generally toxic. Or you can have targeted treatments that will target something like a protein or something that's quite integral to the cancer, or we signal to the immune system that it's unnatural. They tend to work better, but there's more cases of relapse after treatments, so they tend to be used in combination.
Sure, I'll ask you to come here and give my talk. But basically, so it's true. So the answer was there are many systemic drug therapies, different drugs. We have the traditional cytotoxic drugs that interfere with general processes in cells. They cause DNA damage. The earliest cytotoxic drugs were actually derived from war gas. They were derivatives of mustard gas. And that's as brutal as it sounds, and you have all seen these pictures and so on. These drugs are very toxic, have dramatic side effects. In the meantime, we also have more specific drugs that target more specifically cancer-specific changes, abnormalities, whatever you want to call them. But the systemic therapies cure only very few patients. So what other kinds of cancer therapy are you aware of? What is the oldest type of cancer therapy? Surgery, yes. People have done that. Since people have started writing things down, they have written about the same thing. How you get high or drunk, that's a very important topic. Ever since people started writing, a desire to have general anesthesia, because it's very unpleasant if you need an amputation and you don't have that. Complaining about the next generation that is really useless and gets it all wrong after we have made the world such a great place. In particular, my generation has done a good job. You will thank us for that in the future. We have left you in a perfect situation, and now we start drilling in the North Sea again. That will make your life even better in the future. But still, every generation has complained about the next one, and since the very first documents, people have also tried to cut out cancer. Cancer surgery, the Egypts tried that. It's a very, very old thing, and it's still, and obviously it used to be before general anesthesia was developed in the middle of the 19th century. It's not that long ago, actually. Oh, it sounds very long for you, but the older you get, the more you think, oh, these things weren't actually that long ago. It was really very painful and very awful, not only for the patient, but also for the surgeons, performing a surgery with a patient who's not very compliant. It's not a nice thing. But still, surgery is responsible for the vast majority of cures that we have today. And a good example for that, to illustrate that, is the cancer. I hope you know what sunscreen is. It's called melanoma. Yeah, so I have some moles here. I have a mole. I always show that mole. You can hardly see it from there. It doesn't matter. You know what a mole is, right? And I know already, unfortunately, that this mole has the first 1 or 2 cancerous mutations. It's not very comfortable, but still most of the moles do not develop into a melanoma. And that is a very well observed one. I gave my last presentation where I said that yesterday, so I always look at that one. That's probably not going to kill me. But if it starts to change in shape, in color, whatever, I will go to some surgeon to let them cut it out. Now, if I'm lucky it's not even cancer, it just looks funny, it was not even cancer. It can be cancer, but it hasn't metastasized, it hasn't spread, then I'm cured. And that is how most, and we have cure rates of 80–90% for melanoma, but they are largely for early detection. So cancers are cured by early detection and surgery normally. There can be some adjuvant therapy on top of that, there can be some radiation on top of that, but that's the main way in which cancers are cured. If I'm unlucky and this wasn't the only melanoma tumor in my body, then the likelihood that I give a similar talk in 5 years' time is down to somewhere around 10–20%. What is a massive improvement in the last 10 years? Treatment of melanoma has strongly increased or improved because we have some immunotherapies. So if I had told you the same thing 5 years ago, I would have said, oh, the chance that I do the same thing in 5 years is rather below 5%. So there has been an improvement. But also immunotherapies come at a cost. And with the immunotherapies that are used to treat melanoma, we use so-called immune checkpoint inhibitors. And that means every immune cell has many different interactions with normal cells to see whether the normal cell or the other cell or whatever it is actually should be there, is in a healthy state, or needs to be eliminated. And what we do, and this is based on a number of activating and inhibitory interactions. And what these immune checkpoint inhibitors do, they remove some of these inhibitory interactions. So the immune system starts to recognize more cells as damaged, wrong, false, whatever you want to call them. And that's actually very good if that means the immune system starts to recognize the cancer cells. And this is how about now 10, 15, maybe 20% of people with metastasized melanoma are probably cured. At least they are given 10, 20 and more extra years. But the downside is, of course, if the immune system starts to recognize something that it shouldn't recognize because these are perfectly healthy cells, and these cells are really important, that has massive side effects. 1% of people who receive these immune checkpoint inhibitors die from an overshooting immune response. 40% develop autoimmune disease. And the issue here generally is, and why basically the answer to the question, or maybe I should ask this question now. So why is it so difficult to cure cancer? What is, if you think about, you have a bacterial infection, you have an antibiotic, that usually works well, although We will leave you a world in which this has also been at least partially messed up. And between having a cancer cell and trying to kill it. What is the difference conceptually?
If you've got like bacteria that's a foreign body, so it's easily recognizing of symptoms, you know, you get sore throat when you go to doctors and, you know, it's bad enough, like, oh here, have this miracle medication that will probably make it go away, whereas if you've got cancer, firstly, everyone's mutations are completely different, so there's always a chance the medication won't work at all, and then on the other hand, at the end of the day, they are cells from your body. They do accumulate mutations and therefore become slightly different from the rest of your cells, obviously behave abnormally. But if you're actually like looking at them like under a microscope, sometimes they can be very hard to recognize because they disguise themselves to hide from the immune system and other things. So you can't just give someone a bog standard single pill that will cure all cancer because it does, you know, there's such a wide variety and it is designed to not be detected by your body, so the reason the medication will be better.
If you want to do a PhD, drop me an email. Yeah, that's correct. I know I don't have much more to say about that. Yes, a bacterium is a completely different organism. It has a cell wall. For example, bacteria have a cell wall, prokaryotes. You probably have learned that in school, I hope you have. Penicillin, for example, targets the formation of the cell wall. We don't have a cell wall. We tolerate tons of penicillin compared to bacteria. With cancer cells, coming back to the mole, these are just the normal cells. They are already in my skin, and they accumulate the one or the other mutation and change. So it's very difficult to find something that is really specific for the cancer cell that makes this small therapeutic window. And this is why all cancer therapies for the foreseeable future will be toxic. Yeah, we may have more or less toxic therapies, but compared to any other therapy, cancer therapies, our tolerance is much higher for toxic, in terms of toxicity for cancer therapies than for any other therapy. I hate rooms in which I talk where there's no clock. So I have 15 more minutes. So yeah, now I jumped a little bit from my slides. It doesn't matter, I guess you get the context. So we need these effective systemic. So if we want to do more, so we can do two things to improve cancer therapies. We can have better early detection, or we can have better prevention. We have actually a lot of prevention. People smoke much less. So if you want to really do something against cancer, don't start smoking. That's the most and simplest thing. Since smoking is decreasing, the number of cancer cases is also decreasing. Smoking is the single most dangerous thing you can do. On the upside, if you smoke anyway, you can do anything else. Yeah? So not smoking is really, so prevention is a good thing. Then early diagnosis and early therapy is a good thing, but it's much more difficult than it sounds. But if we have people who are diagnosed with cancer at an advanced stage, we need effective systemic therapies, and that is difficult due to the small therapeutic window that we have just talked about. And that's actually what our research is about. We have cancer patients who we can't cure by local therapy, so we need systemic therapy. And we have for many cancer diseases, you know, you see my beautiful stickman at the left, to the left. For many cancer patients, we initially have a therapy where we have a response. That can be what we see here. One of the tumors disappears, the two others shrink. That is a partial remission. We can have a complete remission. That means there's basically no cancer detectable in a person anymore, but it still comes back at some point. Or we can have stable disease. That means we don't have any growth for a while. But after a while, for the vast majority of cancer patients, the cancer comes back and the cancer cells become resistant, and there's nothing we can do. We don't know what to treat them with, and they're going to die from the disease. It can be more than one therapy cycle, but in the end, the cancer cells become resistant to what's available, or what we know might stand a reasonable chance to be effective, and then we don't know what to do anymore. And so what we would want, or what we would need here, would actually be effective next line therapy. So if we run out of options, we want to know what could be a therapy that actually works now. And that is very complicated because cancer is a very individual disease. That means every cancer is different. If you look around, we all look different. That means we all have a different genetic background. We all live different lives. We may smoke or not. We do other things. We may eat a lot of bacon or not. We may be vegans. We may drink alcohol or not. We may go out in the sun. We may exercise. All these things together define us as individuals. But that also means our genetic makeup, with our lifestyle, with the random events. Cancer is also associated with random events. We could have a whole session about whether cancer is just bad luck or not, and the truth is somewhere in the middle. This all results in every cancer patient in an individual disease. And in order to find out how to treat cancer patients for whom we at the moment don't have the appropriate therapies, we really have to understand the individual cancers better. And for this, we need biomarkers. We need something that we can analyze in the cancer patient, from the cancer cells, whatever, that tells us what kind of therapy such a patient would need. And obviously in an ideal world, but that is really further in the future, we would understand in this patient who has responded and who has some cancer cells left, how we can specifically target these cells. And then we might be even able, in 1, 2, 3 steps, achieve cures in the future. I always forget that I can switch the slides with this thing here. So there is a lot of progress in how we can monitor the evolution of cancer cells in a patient. And this is done by something, somehow, I'm too stupid to, it's not very strong. It works on my hand, but that's not helpful for anyone. We use liquid biopsies. So liquid biopsies is one of these examples that also scientists who are usually very pedantic with many things and have to be pedantic and precise can come up with flawed terms, because there's nothing liquid about these biopsies. The biopsies are, because what we are talking about are largely circulating tumor cells, circulating tumor DNA. In our blood, all sorts of things are flooding around. You might have heard that it is now possible to actually analyze the DNA of an unborn child by taking blood from the pregnant mother. And in the same way, if you have cancer, I hope you don't, there will be circulating cancer cells and circulating tumor DNA that you can extract from the blood. And we can analyze them, and we can learn a lot about how the cancer cells evolve. What we can't do is translate that information into improved therapies. Most of the information that we get out of that, we can't use to help a patient. So we need to understand better the information that we get out of these liquid biopsies, out of circulating tumor cells, circulating DNA. And I think this requires preclinical model system that requires a better understanding of how cancer cells become resistant. And that is important because we need to do this at a systems level, and we can't do this in a patient. We can't get every cancer cell out of a patient. If we could cure them that way, and we wouldn't need all the other stuff. We need functional studies. We need to be able to manipulate the system. And we need to try out therapies that we normally couldn't in a patient. Because, of course, every patient has a right to the best available therapy. And we can't treat patients as guinea pigs. There are many ethical issues. So we need model systems, and then we need a large number of these model systems because every resistance process is different. And that's exactly what we try to do. We take sensitive cancer cells, we take cancer cell lines. If you get a bit of a tumor or leukemia cells and you are lucky, the success rate is low, below 10%, far below 10%. You can establish a cancer cell line. A cancer cell line are cancer cells that you can cultivate in a lab, in a cell culture dish, forever. We think forever. We don't really know because we are not designed as humans for eternal experiments. But the oldest, who knows what is the oldest cancer cell line that ever was established? Ever heard of the oldest cancer cell line? It's quite popular, at least if you follow the media and you know about it, you see it regularly. Yes, HeLa, a cervical cancer cell line established in 1951. And it's still growing well in labs all over the world. So we think they grow forever. So we take these cancer cell lines, we have quite a few of those, and we test them for sensitivity to the drugs that would normally be used to treat a patient with this cancer. And then we adapt the cells to grow in the presence of higher and higher drug concentrations until we have a resistant subline of our cancer cell line. And then we can compare, and when can we identify biomarkers that tell us, that indicate when the drug stops working, what in itself is very important because once anti-cancer drugs, they are all toxic, stops working, they are just toxic. So just sparing non-effective toxic drugs for a patient is a big step forward. And because we have our model of resistance, we can test drugs, we can identify new therapeutic targets in these resistant cells. And ideally, we then have a combination of a biomarker and the next line drug. And if we have this understanding, these hypotheses, we can go into the clinics and can see whether we can help patients whom nobody can help at the moment. You need many models of resistance to be able to do that. And to do this, we have established something that is called the Resistant Cancer Cell Line Collection. That's a collection of 2,500 cancer cell lines that have been made resistant to different drugs. And we analyze them to find these biomarkers that help us to develop new anti-cancer therapies. Do we have any? We have 3 minutes left. Yeah. Normally, I would now show you an example where this really works in the clinic. So I'm not just talking a lot of nonsense to you, but that actually works and that we actually find things that are relevant in the clinics and can improve cancer therapies. But since we have only 3 minutes left, you just have to believe me or drop me an email to learn more, or whatever you want to do. And But we can answer a few more last-minute questions if you have them. Yes?
Do you think using small molecules to prevent metastasis by targeting proteins that would ordinarily allow the cancer cells to move from one place to another is an effective treatment? Because I know it's used sometimes in prostate cancer to regulate the release of hormones currently, which have like side effects such as osteoarthritis and things, and can cause weak bones, which is very dangerous. So do you think that would be a more effective way of treating cancer?
I think so. This is an interesting one, and that's really intensively discussed. I personally, I'm not convinced that anti-metastatic drugs that prevent metastasis, are our way to cures. And that's very simple because, and the reason is very simple, because the patients who we struggle with already have a metastasized cancer. So the cancer has already metastasized. The metastases are already there. Anti-metastatic drug might play a role in prolonging survival. They might play a role in if you cut out a primary tumor and you're not sure whether there are still some cells, you might increase the cure rates. But conceptually, fundamentally, anti-metastatic drugs are not a way to cures, I think.
Is it possible to use surgery, even once metastasis has occurred, numerous surgeries to get rid of the cancer?
You can use, so you can't really, once the cancer has metastasized, surgery won't cure a patient anymore, because then you have too many metastases in different parts of the organism. But of course, surgery can still be performed in cancer patients. If there is a certain metastasis that causes particular damage, that is life-threatening, that might be removed by surgery, or you might use radiation. So often with brain metastasis, you use and things can happen. People lose speech, and that can be just one metastasis, and you can irradiate it, and then people can speak again for, let's say, 3 months or so. You have to. It's very important to understand that with many cancer cases and with cancer therapy, we are really. It's like warfare in the trenches. Progress is slow, tough, little steps. Yeah, often when you read in the newspapers, you don't read newspapers, right? Wherever you get your news, and I always tell my children they should read newspapers, and they look at me, oh, you're really an old man, Dad. What are you telling us? But actually, newspapers are not that bad. But when you read there, oh, a new drug, it's huge progress for this cancer type. When you really look into it, often it's 6 months or so. Obviously, 6 months are great if they are for me, but it's not really a step towards a cure.
What exactly are biomarkers?
Pardon?
What exactly are biomarkers?
Oh, it can be certain, it can be different things. It can be, the easiest thing is if you find a mutation that tells you something, yeah? Just a change in the DNA where you know, so for many anti-cancer drugs, many of them target certain proteins, and then one mutation sometimes can change the protein in a way that it can still do what it's meant to do, but the drug can't bind anymore. That's the simplest thing. Sometimes it is that you can compare just also the levels of a certain protein in cancer cells compared to in the cancer cells. So if it is high, you know these cells will probably be resistant. If it's low, it's not. So it can be different things, but practically it's mostly understanding mutation profiles in cancer cell DNA.
You mentioned earlier about people being able to be like genetically disposed to certain cancers. How would you determine that somebody's genetically disposed?
Oh, yeah, that's a very good question. So there are cancer syndromes where we know. Obviously, the most famous one are the BRCA gene mutations, the Angelina Jolie mutations that predispose women to ovarian and breast cancer and men to prostate cancer. But for most people, we don't really know. We know that cancer runs in families. Actually, other diseases also run in families, and there's probably a genetic background. And often we don't know that. Yeah, and that's because it's multifactorial. It's very complicated. It's a combination of different things. But we understand this better and better. Yeah, but also here, it's very complicated. Progress is slow. We can't take people apart as we would like to do as researchers, but not as humans. So it is, if it's not very obvious, it's very difficult.
One more question and we'll have to move over.
Do you think that research into improved DNA repair mechanisms, thinking of like studies into Peter's paradox in Wales, which don't get cancer as much as they should because of good DNA repair mechanisms, do you think that that could potentially yield some preventative measures that would actually be useful in preventing more common types of cancer that come from aging and, you know, losing effectiveness of the recombinance of DNA?
Obviously, a lot of research is done in this area, DNA damage repair. It's difficult to speculate how far we get with it. It's also a double-edged sword because cancer cells themselves can increase the DNA repair as a resistance mechanism against therapies that damage the DNA. So everything, it can go in different ways. It is an interesting question. There are new drugs coming that are involved in, usually they interfere with DNA damage repair because the cancer cells are already damaged, and then the idea is you push them this little bit further and normal cells will just be able to cope with that. But how far this will get in take us towards cures and so on, that's very difficult to